and I will be starting us off today talking about experimental design and simple tests. But just a little bit about me. I'm a biostatistician by trade, so talking about like biostatistics, statistics, some kind of R data visualization, and R package development. So some of the things that we're going to be talking about today in this talk are discussing the difference between observational studies and experimental studies, just some kind of basic terminology that are used in experimental design, common experimental designs, some simple statistical tests, as well as some non-standard experimental designs, depending on how much time we have left. So the first thing, and I think one of the most overarching distinctions of this presentation, is discussing the difference between observational studies and experiments. So observational studies are going to be things where we're having conditions that are influencing the response that we as the investigator are not necessarily have any control over. This could be things like maybe we're looking at kind of the phases of the moon or something. We have no control over that. So that would be an observational study. And because of that, it means that we can't randomly assign treatments, and it could lead to some potential confounding. And because of all of these kind of issues that we can't necessarily control, we aren't able to infer any cause and effect relationship. On the other hand, though, we have randomized experiments, and that's kind of going to be what this whole talk is about. And with randomized experiments, we are able to infer that cause and effect relationship. And I did put a little asterisk on that sentence, and that's because it has to be a well-designed experiment. So there's a lot of nuance when it comes to creating an experiment that we are able to infer that cause and effect relationship. And I hope that kind of throughout the rest of this talk, we're able to kind of go into those nuances. So there are four kind of general steps that happen when we are creating an experimental design. The first thing is that we need to choose the biological system. This is things like balancing the physiological relevance, reproducibility, and also kind of determining how complex we need the data to be. The next thing is then determining the hypotheses that are going to be tested. This is things kind of like determining the treatments that we'll be applying, what are the time points, if any. And then the third step is selecting the data to be generated or collected, and then determining the number of replicates. And I've noticed that already in this like past 3 slides, I've gone over some terminology and kind of keywords. So I think it's important to stop now and kind of go over just kind of what some of those definitions are. So the first thing is the treatment, and that's going to be the procedures being studied. If we are having an experiment and we're trying to test if one drug is better than the other, those 2 drugs are going to be the treatment being applied. The experimental unit is the unit to which the treatment is being applied and that we wish to make an inference on. In comparison, we also have an observational unit. They seem a little similar, but they are a little different because the observational unit is the unit on which the measurement is taken. So kind of one example that you can think of is that if you're working with an oven and kind of wanting to test the efficacy of the oven, but the way that you're measuring it is by the batch of cookies that you're kind of using it for, then the experimental unit is the oven, but the observational unit is the cookie, because that's what we're measuring. Oftentimes, they are the same thing, so it doesn't end up being overall too complicated, but there are those sometimes distinctions that are important to make. And the last thing is then the response, and that's the outcome being measured. So then one thing to note here is that, when we're looking at the observational unit, experimental unit to observational unit, if we're working with a 1-to-1 mapping, we get to use standard statistics, which is nice because that's kind of just an easier, simpler route. It's when we don't have that 1-to-1 mapping that we have to start going about doing more of those non-standard statistics. And then just kind of a few last pieces of terminology. Randomization is how we are assigning treatments to experimental units in a probabilistic manner. The control is going to be that baseline treatment. So if you're thinking of clinical trials, it might be the placebo that the patient's receiving, as well as then confounding. And that's when one factor's effect can't be distinguished from another factor's effect on the response. And so the goal of all of this is to reduce that confounding. So the major components of that experimental design, then we first start with the treatment structure. And so that's going to be the treatments that are going to be studied. Then we move on to the design structure, which is all about grouping those experimental units into uniform blocks. And then finally, we have to apply some form of randomization. And this is to enable us to avoid any type of systematic bias. The good thing is that this doesn't have to be complicated. It can be a relatively simple randomization strategy, but it does need to be applied in a manner that avoids that systematic bias. And the reason that we have to do randomization is that we might not know what factors are confounding. So this is kind of enables us to be able to remove some of that confounding even when we might not know where it's occurring. So the first thing we're going to talk about then is those treatment structures. So one of the major types of treatment structures is called the one-way design or a one-way ANOVA. And basically a one-way ANOVA is when we are trying to look at one single type of factor. So in this case, if we're running the experiment, the question of interest is, do these mock samples have any difference between those of the viral samples? Because we're only looking at one kind of factor here, which is viral strain, which has then two different kind of levels, either mock or virus, we can do a one-way design. In contrast, there's also considered two-way designs or a two-way ANOVA. And that's when we're looking at then two different factors. Here in this situation, we have crop, which has two levels, as well as then location, which has two levels. So if we were going to run this experiment, we'd be looking at 4 total combinations, either crop 1 location A, crop 1 location B, crop 2 location A, or crop 2 location. And one thing to note is that in theory, you can have an n-way ANOVA, as many factors as possible, but that's not necessarily a good idea. And that's because we live in a world that has finite resources, be it time, money, and also statistical power. And so it becomes kind of this balancing act of how many questions can we ask and how many different parameters can we include, while also then making sure that what we're doing is a plausibly feasible experiment. So now that we have those treatment structures, we kind of know kind of the overarching questions that we're asking. The next step is, how are we going to put this into an experimental design? And that's where the design structures come into place. Some common design structures include completely randomized designs, randomized complete block designs, incomplete block designs, as well as Latin squares. So the first one of those 4 is the completely randomized design. So this is when treatments are applied randomly to experimental units. This is probably the simplest way to apply randomization. It just means that if in this case we have 4 different colors which represent treatments, and it means that each circle has about a 25% chance of being applied to any of those 4 different colors. There's not really any more kind of underlying mechanism behind it. However, all of these other design structures account for a thing called blocking. And blocking is when we create sets of groups prior to running the experiment. And the reason that we include blocking is because it allows us to reduce variance. And this is because the units within the same block are expected to have similar responses in contrast to units not in the same block. And as mentioned, this is used in many different design structures. So kind of one example of thinking about this is that if we are applying or testing a drug on patients, and we think that there is a very likely case that it might impact males differently than females, we might want to block sex, so that way we're able to account for some of that variance. Just because if we already know that that might be an impact, we can kind of correct for that confounding prior to it even happening. So one of those examples then is the randomized complete block design. And this is where we block those experimental units into those similar traits, be it age, sex, or anything that might be relevant to the study that you are researching. And then the different treatments are then tested randomly and randomly assigned to units within that block. So perhaps in this case, because there's 5 blocks, maybe we're looking at different age categories, maybe people in their 20s, 30s, 40s, 50s, and 60s. So once we've already blocked for age, then we randomly assign the treatments of A, B, C, and D into each of those different age categories. One thing to note is that it also can be an incomplete block design. So perhaps we don't have enough resources that every single one of those age categories gets all 4 treatments. We could make it so that we randomly assign maybe 2 or 3 of those treatments to each age category, and we would just have to randomly assign kind of which treatment gets left out of which age categories. So it's not perfect, but it is a way to kind of allow for that kind of finite resources that is very common and prevalent when it comes to experimental design. In contrast, then we also have the Latin square design, which is more restrictive than the randomized complete block because it's allowing you to block on 2 variables. So then the total number of blocks is going to be the number of treatments. And each treatment is going to appear only once in each row and in each column, kind of like a Sudoku. So it kind of becomes a little bit more complicated, and it has to be, in this case, a perfect square. You aren't able to do like an incomplete Latin design as much. So now that we have kind of the questions that we're asking, we kind of know how we want to set up the experiment. The next question is, how many samples do we actually need? And the reason that this is important is because replication is essential for statistical significance in an analysis. If we increase the number of replicates, it's to guard against the loss of statistical significance due to sample variation and any other technical problems that might arise. But as mentioned before, kind of with that N-way ANOVA situation, this is always a kind of a balance between the cost of how much it's going to have on the experimental design, as well as in the quality of the data that we're going to be working with. Because if we don't have enough samples, it's going to lead to the situation where our statistics are just not going to mean much. But if we then spend too much money and we have more samples than necessary, we've just wasted a lot of money on something that wasn't necessary. One thing to also mention when talking about how many samples is to mention the difference in the type of replicates that we can be using. There is technical replicates and there's also biological replicates. The biological replicates are going to be biologically distinct samples. And this is kind of to test anything with regards to biological variation. So if we were doing perhaps like blood samples, a biological replicate would be taking multiple blood samples from different individuals. In contrast, we could also be looking at technical replicates, and that is when we are doing repeated measures. In this case, if we were doing blood samples, it would be taking blood sample measurements from the exact same individual. And this would be more to kind of look for variation when it comes to things like the equipment or the protocol. So that's another question to take into account when you're looking at how many samples is, are we looking at biological replicates that we want to include, or is it more technical replicates? And one thing to note is that when you are kind of in this question of asking how many samples to include, it's important to potentially consider adding more replicates to the group instead of adding more groups to the experiment. So if you're looking at an experiment with, as we saw with the one-way ANOVA, mock versus viral strain, we might think it might be really useful to add maybe even a third option, maybe drug B or something. But that might not be the best case if we're really wanting to improve the actual statistical power of what we're analyzing. It might be better instead to just simply add more samples to both the viral strain and to the mock samples instead of adding a completely third new group. And I've been mentioning this word statistical power, and I promise that we'll talk about it a little bit more in depth, but it is the quantity typically calculated to help us determine the number of replicates that will be run. And so, as we kind of talked a little bit on the previous slide, the big question of how many samples that follows then is why bother? And the reason is because we want to have scientifically meaning effect with regard to our data. Whether it means that the data isn't significant or it is significant, we want it to actually be a valid design that we've conducted. And we want to be confident that what we've found does match reality. And the big question with that then, as we mentioned before, is that money. That undersized study is going to waste resources by not having the capacity to produce useful results, but that oversized study is going to use more resources than necessary. We also have to take this into account, especially when we're looking at kind of like when we're dealing with humans, is that ethical issue that if we're going to be testing things, we want to make sure that we're not having too many people in the sample if we're not sure even if if the study is going to yield positive results. As well as that grant reviewers are simply looking for sample size and power calculations. And in order to get that money for your experiment, you need to know how to appease them. So then the next task comes at looking at like, what are the hypotheses? We already have kind of talked about like what the general question is. Like with that one-way ANOVA, we know that we're looking at mock versus viral strains. However, when it comes to statistics, we really need to be able to clearly define what those questions are. And those boil down into what the null and alternative hypotheses are. The null hypothesis is going to be what we assume to be true. It's kind of considered the baseline that if we didn't run this experiment, this is kind of what we assume that the world would kind of just think as is normal. It's also denoted as H_0. And basically, with most null hypotheses, it's going to be a sentence similar to there is no difference between. In contrast, then, the alternative hypothesis is what we want to show, and notated as H_A. And it's usually a sentence similar along the lines of there is a difference between treatments. And kind of similar to court, we kind of just assume this innocent until proven guilty. We always assume that the null hypothesis is going to be true unless we have enough favor and evidence of the alternative hypothesis. So kind of another way to state this, putting it a little bit into more like mathematical formulas, is that if we are looking with a population parameter P, we can run a couple of different hypotheses tests. The first one is considered a two-sided test, which basically means that if we're looking at the null hypothesis, we're looking at this population parameter is equal to some baseline statistic that would be context dependent. Whereas the alternative hypothesis is simply that this population parameter is not equal to that kind of baseline value. So it means that we don't care if the value is greater than or less than, it's just not equal. In contrast, though, we have one-sided tests as well, where then we start asking the question, is the population parameter greater than or equal to the test statistic? And we think that it's actually going to be less than. Or we could do the opposite, where the null hypothesis is that it's less than or equal to this baseline value, the alternative being that it's greater than. So in that case, the direction of how things are moving is important to consider. And one thing to note is that the equal sign is always included in the null hypothesis, and that the null and alternative hypothesis must contradict each other. So between the null hypothesis and the alternative hypothesis, we have to be including every single possible scenario. So once we have those null hypotheses, the question becomes then, how can we actually determine which one is the one that we're going to select? Are we going to reject the null hypothesis, or are we going to fail to reject it? And that is based off of the p-value in most situations. And the p-value is going to be the probability that an observed outcome is due to chance. A high p-value means that it is more likely due to chance, whereas a low p-value means that it is less likely to be due to chance. And in most statistics, that default value for the p-value is roughly set to be 0.05, though it is context specific. So perhaps we're looking at more exploratory data. We don't need to be as confident with our results. It's just kind of to see if we need to do further experiments. Then we might set that p-value to be a little bit higher. It could be 0.1 perhaps. But if we're looking at something where we need to be very sure of our results, we might set that p-value to be even smaller, like 0.01. But regardless of whatever you set that p-value to be, if it's 0.05 and if we find that our p-value is less than 0.05, then that means we're going to fail to reject the null hypothesis. Or if the p-value is greater than 0.05, we fail to reject the null hypothesis. We find that there is not sufficient evidence to conclude that the alternative hypothesis that we're looking at is correct. Whereas if the p-value is less than 0.05, which means that it's less likely to be due to chance, that's when we're going to reject the null hypothesis. We do find that there's sufficient evidence to reject the null hypothesis, that there's no difference, and we can start acting as though, and we can start looking more at those differences that we were looking for. One thing to note is that you never actually accept the null hypothesis. But just because the p-value is less than 0.05 or greater than 0.05 doesn't actually mean that's reality. It's just what we found in our experiment. And so that means that we can also run into errors with our hypothesis testing. And so rejecting that null hypothesis in favor of the alternative, it doesn't guarantee that what we're doing is correct, and it doesn't mean the alternative is true despite that very strong evidence. And for that reason, we've also had to take into account that there are different errors that we can. So I think that Eva yesterday showed a similar graph when it comes to machine learning, but when it comes to statistics, we have a similar issue where we have the truth, which could either be the null hypothesis or the alternative, but then we also have the decision that we made, either to fail to reject the null hypothesis or to reject it. So if the baseline is that there is no difference, and that's the decision we made, that's a correct decision. And similarly, if we reject the null and that was the correct decision, that is what's considered power. The other two corners are the ones that we unfortunately want to avoid, but are sometimes unavoidable. And that's where we have either a type 1 error, which is when the null hypothesis is true, but we still rejected it. And that is considered a type 1 error. It's a false positive, and it's labeled as alpha, which is also that p-value typically with that 0.05. We also then have the alternative hypothesis when we find that to be the truth, but we fail to reject the null hypothesis, which is a type 2 error and is also denoted as a false negative. So by construction, the hypothesis testing that we were doing limits that rate of the type 1 error, the false positives, to that significance level alpha, which is the p-value 0.05 typically. And because we kind of select that p-value to be 0.05, we're already choosing ahead of time what that significance level is. One thing to note with that is that with multiple testing on kind of different attributes of the same data, we're going to have to require an adjustment though, in order to preserve that significance level. So for example, if we are looking at data with salmon and we are testing for multiple different types of chemicals, not just like one single chemical within the salmon, we're going to have to require an adjustment because the more tests that we're doing, the more likely we are to find a false positive. So this running correction on the data is kind of a way to avoid that likelihood of finding that false positive. In contrast, though, we have the type 2 error rate, also known as beta, which is a function of sample size, the significance level, as well as the effect size. And one thing to note when it comes to type 1 and type 2 errors is that there is a trade-off and an inverse relationship. So if we try to decrease the type 1 error rate, it means that the type 2 error rate is going to increase and vice versa. So it's all about finding that balance between the two. So as we kind of mentioned before, statistical power is the probability of rejecting the null hypothesis when the alternative hypothesis is true. And as we talked about before, with beta being the type 2 error, statistical power then becomes 1 minus that type 2 error. So when we're trying to calculate the statistical power, it's based off of 4 different elements: the type 1 error, the effect size, the variability, as well as the sample size. And all of those 4 elements contribute into calculating what is the statistical power of our experiment. And as mentioned before, there is that trade-off and inverse relationship between type 1 and type 2 error, which means that there becomes a direct relationship between the type 1 error and power. So a lower type 1 error is going to lower the power, holding all other factors constant. And some typical values for that type 1 error, which are the p-value, are things like 0.05 and 0.1, and it is heavily domain dependent. And then the effect size and the variability kind of work together in that the effect size is the desired detectable difference if a difference exists. So one way that I kind of like to think about the effect size is if you were kind of calculating races. If you were to try and measure 2 different individuals running a mile and you found that there was a 1-minute difference between the two, that would be a very stark difference. But if you were to take those 2 same individuals and then find that there was only a 1-minute difference when running a marathon, you'd find that that was much more negligible. So that's where the effect size really comes into play, and why it is so domain specific is that if you take that 1-minute difference, you might think it'd be the same regardless, but it has to take into account kind of all the other contextual information that you're working. And then we also have the variability, which in this case is going to be the variability of the parameter being estimated. If we're evaluating the difference in means, that means that the variance of the values are going to be coming from the same group. And so that ratio of the effect size and the variability is going to be what determines power alongside those other things that we've been talking about. If we have more variability, we're going to have less power. And if we're going to have a smaller effect size, we're also going to have less power. And so when it comes to that typical value, as kind of mentioned with that kind of like mile versus marathon example, it's going to be determined by the data that we're working with and also the domain knowledge. So if you are working with statisticians, when it comes to kind of calculating the power that you want to be working with, that will be kind of where it's really heavily important to have that kind of collaboration where either the chemists or the biologists are able to kind of state what they think a good effect size would be. So then the statisticians can then kind of look at the equations and be able to plug in those values. So it's definitely a very collaborative effort, as with all of science. And so usually some of the quantities that we want to estimate are things like, if I have X number of samples, what is my power? or if I have so many samples do I need in order to get to an 80% power. And most domains, we only have one estimate of variability, which leads to only one power calculation, which makes things pretty simple. However, with biology, especially with omics data, we often have multiple response variables, which leads to multiple power calculations. And one thing to note is that we're not really going to dive into kind of all of the actual equations and things, just because thanks to today's technology, we have computers to do that. So highly recommend. I often use R in order to kind of calculate sample sizes and things. So that's kind of like one thing to take into note, is that it's important to have the theory, but thankfully we also do have the computers to do a lot of the legwork when we need. So now that we kind of asked all of those questions in terms of how many samples do we need, how are we going to structure the data, then we get to the fun part, which is analyzing the results after the experiment has been conducted. And that's where we get into simple tests. And with simple tests, or standard tests, we often have two main questions, which are running quantitative tests or running qualitative tests. And quantitative tests are simply asking, are there differences in the mean abundances of each biomolecule between the treatment or groups? In contrast, we would have then qualitative tests, which are there any patterns of presence and absence for each biomolecule associated with each group? So for example, if we are looking at biomolecule A, we might note that we want to see if these values, 16.4, 16.9, 0.2, and 0.7, is there any statistical difference between those values from the green group versus those in the bluish purple group? And that would be a quantitative test. But if we were to look at molecule B, we might notice that there are no observed values for the green group, which means that we can't really run a quantitative test. But we're still not kind of completely out of luck, because we could look at a qualitative test and perhaps run something to observe, are there any statistical difference in the number of observed values in the green groups versus those in that purple-blue groups? So we're first going to start talking with quantitative tests. And that usually is where we kind of look at ANOVA, or the analysis of variance, which utilizes the F-test. And so the F-test null hypothesis is going to be asking, is there a difference between, if we have, in this case, three groups, the mean of group A equal to the mean of group B equal to the mean of group C? And the alternative hypothesis is simply going to be answering the question that at least one of those groups is different. So groups A and B could be the same, but if the mean of group C is different, we would then reject the null hypothesis. After which we can then also run some post hoc pairwise comparisons, which then allows us to kind of dive deeper into those kind of comparisons that if we found that C is different, we can still ask the question, is A and B different from each other? And then we would have then, in this case, three kind of sub questions of, is A and B equal to each other, A and C equal to each other, and B and C equal to each other? And for all of those post-hoc comparisons, we would then be able to calculate a p-value, as well as then the log fold change, which is considered to be the log of the mean of group A over the mean of group B, or whatever groups that you are specifically looking at for that specific pairwise comparison. So when we're thinking about the assumptions of kind of the ANOVA testing, and for these quantitative tests, we have three main assumptions. And the first one is that the data is independent. We are assuming that there isn't any correlation between like the different samples that we're working with, that they are all just independent from each other. We are also assuming that the data has a constant variance, and that the data is normally distributed. If the assumptions are not met, we're not completely out of luck. We have a few things that we can do. We can potentially transform the data using something like a Box-Cox transformation, as well as it's also important to note that not all of these assumptions are held to the same magnitude. So for example, if the data is not independent, then you really can't run these ANOVA tests. That is an important criteria that needs to be met. However, with like the normality assumption, we are allowed a little bit of wiggle room. If the data is not perfectly normal, but it's kind of close, we might be able to make a good judgment call and determine whether or not it's okay to run still with ANOVA and just kind of the standard statistics. So that's another case where it's important to kind of talk with the statistician to determine just kind of how much the data is violating assumptions. Additionally, it's also important to take into account any outliers that you might find. And outliers are going to be when the response is extreme in comparison to the other responses with similar predictors. And a lot of times the quick thing that we want to do with outliers is to just remove them because they look weird. And while there might be a valid reason to remove outliers, it's also important to use caution because that outlier might be an important part of the story that we're trying to tell when it comes to the statistical analysis. So just because it doesn't necessarily seem to make sense, it doesn't mean we should remove it. We should only remove outliers when we have kind of a strong reason for doing so. Like we know that the experiment was completely batched when it came to running that sample. And then we're like, oh, we see this as an outlier. That makes a lot of sense. We should probably remove it from our samples. And we had already touched on this a little bit, but I wanted to dive into it a bit more, which is multiple comparisons. So that concept of when we are looking at, like, for example, with that salmon, we were testing multiple different chemicals at once. And so we needed to correct for that p-value adjustment. And this is, again, based off of the type 1 and type 2 error rates. And we want to reduce the type 1 error rate with multiple comparison analysis. Because if you are running many tests, it's likely that something will be found significant at that 5% threshold, even if it isn't. And I like to think of this, I think there's a book called The Law of Improbability. And with that, they talk about this idea that basically that the more things happen, the more things are going to happen. So if you are constantly running tests, then it's very obvious that at some point you might find something to be significant. And it doesn't mean that it actually is, it's just by chance, because you ran the experiment so many times, something popped up as significant. And that's what we're trying to correct for here. And so there's a lot of ways that we can correct for it, depending on the data that you're working with. There's the Bonferroni method, Tukey, Dunnett, among a plethora of others. And the thing to note here is that there is no best procedure, just the best one for your situation. Whether you're working with exploratory data, confirmatory data, maybe you're working with comparisons to control, or you're just looking at all pairwise comparisons. Some of these do need to be pre-planned, but others can be done post hoc. And so a little bit more kind of about the main ones that we at least use in our lab are the Bonferroni method, which can be used for both quantitative and qualitative analyses, as well as for both case versus control and for all pairwise. And the same can be said about Holm method. Whereas if we're looking at the Dunnett test, that would be if we are looking at only the comparisons between case and control, and can only be used for ANOVA or those quantitative tests. And the Tukey, which can be used for then all pairwise comparisons, but again, can only be used for the quantitative test. Now that we've talked about kind of those quantitative tests and kind of what we do when we are looking at perhaps like the difference between mean log abundances between different groups of samples, we can also dive into then the qualitative tests, which are kind of when we don't have enough observations to conduct a quantitative one. So we could then determine if the proportion of missing values associated with each treatment group are then compared to random chance. So if we look here, we might have treatment A, which has then 1 present value and 4 absent. And we might want to know, is this kind of distinct, if this kind of ratio, is that statistically different from that of treatment B, which has 3 present and 1 absent? So that's where the G test comes into play, and it helps us to determine if that proportion of missing values are associated with the treatment group compared to random chance. If we're working with a very, very small size of samples, we could also use the Fisher's test of independence. So one thing to note with the G test is that we do have a couple of requirements. The first thing is that we need to have the number of samples to be greater than or equal to 3 replicates per treatment. And this is just because if you're looking and you only have 1 or 2 samples per group, then testing whether 1 is different than 2 observed values doesn't tell us much. So we do need to have at least enough replicates to be able to conduct this test. Additionally, it's not typically applicable for metabolomics data, and that's just because with metabolomics, not often that much missing values. So you're just kind of running it and find a lot of very not significant p-values. So you could run it, it's just probably not going to be very useful. In comparison, it isn't applicable for isobaric labeled proteomics data. And that's just because of the way that missing values are kind of found within isobarically labeled proteomics data. So if we look at kind of the label free, we notice that there doesn't seem to be any kind of like pattern with where observations are being found or not found. Whereas if we notice here on the I track, which is isobaric labeled proteomics data, if you have kind of a missing value, it's likely to be missing for almost the entire group or then even all samples. And if it's observed, it's going to be in almost all of them, or at least within each group. So because of just the way that missing values are often observed and not observed in isobarically labeled proteomics data, it doesn't make sense to run AG. But we also can use both the quantitative and the qualitative. So we can run ANOVA and G-test to kind of try and cover our bases. If we, for example, in these data sets, we had enough samples. We had 78 samples to be able to run a quantitative and 446 to run the G-test, and there were 17 that were able to run both. In comparison with the plasma data set in this example, there were 102 that could be ran using ANOVA, 25 using the G-test, and none that were found to be able to be used both. So this is kind of a way that we're able to look at kind of the entire picture of the experimental design. We can, if we run both the quantitative and qualitative, we can kind of see where those quantitative analyses are possible, where the qualitative are, and also be able to kind of identify where is it even that both are applicable, because on the surface, we might not notice that, like with this BALF data set, that there's 17 that can be ran with both, whereas with the plasma, there were 0. So that could also be an interesting thing to know when running these experiments. So then we also have kind of non-standard experimental design. And the big thing that we want to mention with this is that you should seek a statistician when working with especially non-standard experimental design. And this is because it's very important to kind of have that background before you even start planning your experiment. Because if you have those conversations with your statistician as early on as possible, it will just make the entire running the design as well as analyzing the data as smooth as possible. If you just kind of start the design and then it turns out that the data kind of has this kind of non-standard statistical approach to it, it might mean that there were elements that needed to be considered prior to running the experiment, and now it's too late and the statistician can't really do anything with it, and the whole experiment is kind of a waste at that point. So it's really important, especially when you are going to be working with some of these examples that we'll talk about, to like really seek a statistician right away. So that would be things like when we're working with non-independence, especially. So that would be looking at things time course, which could be repeated measures. So for example, one case would be if you're working with like mouse litters, we would expect that all of the mice from like one litter more closely related to each other than mice from another litter. And so that's how it would be in terms of this correlated data and repeated measures, as well as then numerous samples from one soil core, as well as if the data is not normally distributed, working with things like censored data, which would then lead to a type of survival analysis. So then some type of non-standard designs include things like a paired study, where if we're looking at like a before and after, we'd be then looking at a paired statistic, where if we were measuring perhaps a leaf, and then we applied some type of treatment, and then we looked at the exact same leaf after the fact to see if there were any changes, and we did that for multiple leaves, that would be a paired study. We also be looking at a longitudinal analysis. So for in this case, we might be looking at the log abundance of kind of the age of children at some point. So we might be studying that same individual at their age 1, 2, and so forth. So because that same individual is being measured at all of those different time points, we expect their values to be more correlated with their own like values than with any of the other children in that experiment. So overall, the main takeaways of this presentation is that experiments are only as valid as their design. And an experiment with a proper design can help us to detect that cause and effect relationship. Additionally, there are quantitative and qualitative tests for analyses, and that replication is essential for statistical significance. And finally, that you should always seek a statistician before starting an experiment. And I believe that we have a little bit of time, so I'll go through this slide real quick. But just some kind of other considerations to take into account when you're working with an experiment is, like, if you know beforehand that you will be having missing values, like particularly proteomics, which often has a lot of missing values, power calculations are going to assume no missing data. So that's another thing to take into account and kind of talk with your statistician about. The testing with more than 2 groups is going to lead to multiple test correction, as well as then hypotheses not dealing with means, such as like trend analysis over time. Those are going to be things where we have to take into account variability, which isn't a straightforward calculation. So these are all just kind of some other examples of when you're working with some more non-standard statistics. And so just kind of some final tips is, contact your statistician before planning replicates. And then also with regards to groups versus replicates, determining how to allocate those resources. Awesome. And that is all that I had to say. Thank you so much. And yeah, I'd be happy to take any questions.
We have an online question. So the question is, transforming your data could result in a normal distribution. However, it bothers me that the raw data does not follow a normal distribution, and that doing so seems like manipulating your data, which I know is not the case. So how should we think about what a normal distribution is, and why some data follow such a distribution by default, why some do so after transformation, and why some do not, even if you transform the data?
Yeah, that's a very good question. I think that the way that I view normal distribution and normalizing data is more as a way to, as like a technique almost in a way, and that the data needs to be normally distributed because in order to apply these kind of statistical techniques that we need to work with, it works best on kind of that bell-shaped curve. So if you have to transform your data, that's not necessarily data manipulation, because we're simply trying to kind of format the data into a way that makes sense with those statistical techniques that we have. In theory, there are other techniques that we can use for non-normal distributions. It's just that normal distributions have been so widely researched. So I think of it, normalizing, especially when you have to transform it. It's more of a way of kind of formatting your data. So the same way that if you would be kind of formatting your data, if you were going to plot a graph, the way you kind of have it structured might not immediately work. So you just kind of have to format it a little bit. So yeah, I think kind of thinking of normalization more as formatting is a way to remember that it's still a statistically valid thing to do.
Hey, just with regards to like using some of these, like a 2 key or whatever, to correct for where you have like a large chance of false significance versus just lowering the significance threshold, how would you decide which to use in a given context? Do you see what I'm asking? Like, you could just say we need a significance of, 0.00001 because we have so many different so many different chances of finding a spurious correlation, versus you could say we're going to run a correction of some kind. So how do you decide in a given context what you're going to do?
Oh, I see. Okay. So you're saying instead of applying a correction, just make a more conservative p-value?
Yeah, or something in between. You do both. You make like a middle-leaving conservative p-value, and you run some kind of correction. Like, how do you know which to do?
Yeah, I think the big thing with that is that you do want to still keep that p-value at kind of what is the kind of domain specific, kind of context specific area, because there's a reason that it's at that kind of 0.05. So for example, like a 0.05 value for perhaps like an omics experiment is going to be very different than a 0.05 p-value for exploratory data analysis. So by using multiple corrections analysis, you're still able to stay within that domain specific kind of realm. Whereas if you were to make the p-value incredibly small, like 0.0001 or something, you're now kind of changing almost like the domain that you're working with, even though you would probably still potentially yield similar results. But I also think that by changing the p-value, you might be running into the issue of it's unclear how I guess low you would need the p-value to be to keep it comparable. So then you might be skewing the results because you might just be specifying a different p-value that doesn't make sense.
I just also add to that answer, these multiple correction methods are designed to control the error rate over the family of tests, right? So that if you're to arbitrarily just lower that threshold, then there's no guarantee that across your entire set of tests, you're still controlling that type 1 error rate at whatever threshold value, right? So it's important to think of the entire family of tests as far as type 1 error goes, as opposed to any individual test. I think that's the key. Does that make sense? Yeah.
Hi, great talk. I have two questions. One is regarding the normality test that was just discussed before. And so you're saying that when you have a set of data and it doesn't fall into a normal distribution, you think it's better than instead of applying the statistical analysis for the not normal distribution, just to transform it into it falls into a normal distribution and then apply the statistical analysis for that?
So you said that like if you're working with a non-normal distribution.
Yeah. Like, so instead of just applying the statistical analysis for the data that doesn't fall into a normal distribution, do you think it's better for us just to transform the data and to it falls into a normal distribution and then apply the analysis?
Yeah, so I think it really depends. Like I mentioned that the normal distribution tends to be the one that's kind of the most relaxed with regards to assumptions. So I do think that it kind of becomes a holistic decision that like if the data is very clearly not normally distributed, so perhaps it's like bimodal or something, like then you obviously need to apply some type of transformation or just not run standard statistics on it. However, if there's just like a little bit of like a skew or something on one side, that's where you kind of have to play that game of like, it's probably okay, maybe it's not, but that's kind of, yeah, I would say that if the data, it's still an assumption that is important to me. It's just a matter of like how non-normal. So I would say if it's still roughly normal, that's kind of where it might be okay to continue with just standard statistics. It's that question of like, if it's clearly violated, then.
And another question is that about the missing values. For like mostly all mixed data come up with missing values, but those missing values sometimes are important for us because sometimes something is produced or is transcribed in our control, but it's not transcribed in our treatment. So we need to have a way to compare that, but with the missing values, it's just lost because you cannot really apply anything to that. So do you have any ideas on how to overcome that?
Yeah, so I think that's kind of where the qualitative tests, unfortunately, kind of become the best option. And just that if you have missing values, you can't calculate the mean of it. And so it's one of those just like, unfortunately, it is important, but unfortunately, we also just have that limitation of that you can't really run quantitative tests on data that you simply don't have. And so I think that's where we have that G test for the independence of missing data, just to have at least some type of analysis, because you're right, it is important to know where those missing values are and aren't. And that kind of at least is some type of workaround that we can still kind of analyze. Are there any significant trends with at least how the data is missing? So I think, yeah, I think the G test is kind of the best situation that we have in a situation where, unfortunately, it's not great to have missing values.
Thank you.
We have another couple questions from the online folks. We have one asking, are you expected to have a deep understanding of stats in a bioinformatics role in the present day or in the near future?
And I think I'm biased. I think it's really important to at least have a background in statistics. of some type, like obviously not like degrees or anything, but just kind of have some kind of background knowledge when it comes to, especially with things like the standard statistics. But I also don't think that it is expected that everyone is going to be this master of statistics or something. And that's why I hope a big takeaway of this was just to make sure that you, when you're designing experiments, when you are doing analyses, to reach out to those statisticians, because they can like lead you in that correct direction. So I think it's always helpful to have a little bit of context, but I also know that that's not, I know statistics is not everybody's cup of tea. So I think, yeah, just knowing who you can turn to that has that knowledge is going to be essential in bioinformatics and any field.
So, like, often we find that biological data do not adjust to many of the assumptions of the statistical standard tests. Do you think that Bayesian statistics should become more relevant nowadays because of that?
Yeah, I do think that it has its time and place, and I do think that, yeah, with all statistics, it's going to be context dependent. So as you're mentioning that, like a lot of the assumptions aren't necessarily always going to be met, especially with biology data, because biology is such a, there's so much variability within biology. So I do think that it makes sense to have more of a push and change in some of those directions. I do think it's also hard to kind of change what, like 50, 100 years of kind of one type of analysis. So I don't think it will be anything quick by any.
I'll also add that I do think Bayesian statistics are powerful, but it's kind of like changing the problem, right? Because rather than normality, now you have to find some conjugate prior that, you know, you could find a solution for computationally, right? So the problem becomes more complicated, but you do have more flexibility than standard methods. So it's a trade-off, right? But certainly Bayesian methods, non-parametric statistics, permutation tests, these kinds of things that don't maybe rely on distributional assumptions could be helpful, right? Bayesian is all about specifying different probability distributions, so that's kind of separate from this non-parametric stuff. But you know, there's options beyond traditional statistics. But the reason why traditional statistics have staying power is because they work in a large variety of situations, right?
I just wanted to go back to blocking. So say you have a completely randomized block design, and in your statistics you find that the block effect is significant, or, you know, explains a lot of variability in your data. What do you do with that information other than just rest assured that your blocking was sufficient, right? Like, should that inform, you know, the rest of your statistics, or should you do something different with the data because you see such variability across your blocks?
Yeah, I mean, I think that if you see such strong variability within the blocks, so you're saying that like in that example of like that maybe the different age groups, that we noticed that like within the 20-year-old group range, like the response was a lot higher than those in like the 30-year-old, and then even a little bit lower than in like the 40s or something. Is that kind of what you're saying? So I would say that like the first thing would be that if you do kind of find that difference, yeah, then you would be like, yeah, wonderful, like we blocked and there was a reason for doing so. And then I do think that it is then, that could be then something that you would use then to, maybe not particularly with that experiment, but that is something that you could then, that might be an interesting research question then dive deeper into and like down the line. So I do think that the goal, like with blocking and then also just randomization in general, is to reduce that confounding. And so if it's accomplishing that, then like the job is done. So like blocking is kind of a way just to additionally help that kind of being able to reduce confounding.
Right. Damon, you got a lot of questions here. So another online question for you. Let's see. Are there any good resources to go about to learn more about experimental design and statistics relevant to observational studies?
Ooh, with observational studies, I would say with observational studies, a lot of, at least in terms of like, the thing about observational studies is that a lot of statistical analyses can be conducted that you do with experimental design. The issue then becomes in terms of the interpretation, in that you can run ANOVA, you can like run qualitative tests, but then you're still not able to kind of have that cause and effect relationship. It's more of a, there is a strong correlation between this factor and this response. So Javi, I don't know if you're like, if you have any resources on observational studies per se, but I would say a lot of what's being done with experimental design analysis is still similar. It's just in terms of the interpretation.
Yeah, I think that's accurate. You could run the same kinds of tests, but it's just a matter of whether or not you could make that causal inference. That being said, there are methods in stats for causal inference to overcome the fact that you don't have a traditional experiment. And that's a whole kind of field. So I'd encourage you to look up causal inference and look for any number of those books. The Book of Why by Judea Pearl is a nice kind of high level description of causal inference. But beyond that, I don't have any specific that come to mind. All right, I think we'll do a couple more online questions and then we'll get ready for the next speaker. So, okay, so we have somebody saying, I get confused when some statistics references refer to normal distribution as more of an issue of the residuals than the actual data. Could you please expand on this?
So with the normal distribution, I don't think we have any plots on this presentation, but one of the reasons why one of the graphs that people kind of use as the diagnostic for normality is called the QQ plot. And that in theory is going to be just this like diagonal line, and that's based off of the residuals. And so it's kind of one way to be able to better see, especially when you're working with like normal distributions or distributions that might look a little normal but not perfect. It's kind of hard to tell with that kind of bell-shaped curve whether or not that is going to be normally distributed, because you also have other distributions like the T distribution, which kind of look similar but aren't necessarily normal. And that's where then the residual plots like the QQ plot kind of help a bit more, is that it's able to show those tails a bit more and where you're able to see those residuals. So I guess that's kind of one thing that I'm thinking of when it comes to the kind of the issue of residuals with normality. I think in general, it's going to be like the same issue. It's just a matter of being able to kind of better holistically, like, and like visually being able to tell kind of how actually the data is normally distributed, and the residuals is one way that it might be easier to tell and make that judgment call.
I'd also add that typically we're talking about residuals because this is in the context of a regression model. And for the regression model, we're assuming that the error of that model is normally distributed, right? You're modeling your data as a function of your variables, your covariates, and then there's some uncertainty in that relationship, and then that uncertainty should be normally distributed. And so we look at residuals because we're kind of taking out the part that we explained through our covariates and then looking at the resulting error of our model and those errors should be normal according to what we're assuming by fitting a regression. So the last question that we'll take today, and then others will get to after the fact, responding through Discord, is about outlier values. So I often run into outlier values in gene expression data generated by qPCR. 1 out of 4 replicates is often an outlier. Is that reasonable to remove it?
Again, I think it really, outliers is such a weird kind of concept in statistics, and I think it really highlights some of the subjectivity when it comes to statistics. It's not just kind of this like cold hard threshold or something, especially like we have with like p-values and things where it's like 0.051, okay, we fail to reject because it's not less than 0.05. So I think with outliers, there is a little bit of that nuance. So I think it is very heavily context dependent. So if you're only working with 4 samples and one of those is being identified as an outlier, then you're left with only 3 samples, which isn't a lot. So that's where it kind of like, sometimes it might be better to like err on the side of keeping it so that you like maintain the number of samples that you have in the data. At the other hand, if you like know that it's clearly something wrong with like the way that the equipment was being ran or something, then there is a strong case to remove it. So unfortunately, I can't really give you a straight answer for that because it's just, I wasn't there for the experiment to know all of the context and nuance for it. But I do think that it is, I would hesitate to remove it based solely on the fact that there are only 4 samples in each group.